a big welcome to everyone who joined us for this morning's afternoon, for some of you, session that is presented by our Board on Life Sciences. We are really happy to welcome you to this discussion, speakers and audience, on behalf of the board. And briefly, to allow plenty of time for our discussion, the purpose of this presentation, which, as you know, is on innovations and implications of advances in computational analysis in neuroscience research, the official title of the session, subtitle being Unraveling Insights into Brain Function and Plasticity, of which I know we're all very interested. We, in this discussion with our distinguished speakers, are hoping to better understand the current science about biochemical and electrical signals at scale, as well as what we always do to think about the implications of this new science and these advances for the broadest possible impacts, technical, but also societal and policy-related implications. We know that our speaker is going to talk not only about where we are currently, but also about future trends that they anticipate that will bring us very far down the road on better understanding these topics of brain function and plasticity. So the moderator for our presentation, we're very fortunate today to have Dr. Diane DeJulius, who's a distinguished research fellow at the National Defense University. Her background includes a broad experience across not just the National Defense University, where she is doing research on biotechnologies in biodefense, biosecurity, but also a background earlier on as program director at NIH, where she focused on neurosciences research and neurodegenerative diseases. In between then and where she is now at National Defense, she was Deputy Director for Policy and Planning at DHHS, and also served in OSTP as a director in life sciences and behavioral and social sciences. So we are very pleased to have Diane here today to lead this discussion. And so a big welcome to Diane and to the other speakers. If you wouldn't mind, Diane, I'll turn it over to you and ask you to introduce the other speakers.
Great. Well, first of all, let me say thank you so much. And thanks to Kavita for having me and inviting me to this group. I think this is going to be a really robust discussion. And adding on to just a little bit of that introduction already, I'll say that since I got involved in the neurosciences, there's been huge leaps and bounds in advances, and not only in the way we can see and look at the brain and the nervous system, but how we can measure things, how we can do metabolics. We've gotten from very small, sort of looking into the black box, to seeing what's going on in the black box in much more meaningful ways into the nervous system. So thanks very much for inviting me. I think this is going to be a great learning session for myself as well. So the way that we're going to be handling this today is I have a series of questions, and I know that we have a set of individual speakers who are going to be potentially giving short presentations, and alongside with perhaps a handful of slides for each of the individuals. And I know we have at least one person who's going to sort of talk more specifically in the ethical, legal, and social implications of these technologies. So let me just quickly ask Kavita and/or Andrew, should I go in the direction of how I see people on the list, or how do you want me to go? How do you want to get started with the individuals? I can just go down our list of individuals one per. Is that right? Okay, so that would put Dr. Kafui Jizraza first as our first presenter. And I do want to note he has completed a PhD in neurobiology at Duke and the first African-American to do so. His research focuses on understanding changes in the brain in neurological and mental illness. He also has an MD from Duke. Yay, go Duke. I can say that now because I spent time in North Carolina in 2009 and did residency in 2016. It was also on CBS 60 Minutes, some interesting conversations there, and has won numerous prizes and awards in the outstanding work that he's been doing. So I will stop there and turn it over to give the most time to our speaker.
Well, it's a tremendous honor and a pleasure to be here. I see some friends out in the audience. Hi to y'all out there. And I thought I'd talk a little bit about my research and an intersection with a topic that's been sort of top of mind for many of us, and that's artificial intelligence, right? And sort of the merging of what we would call human intelligence, so our quest to understand the brain, and artificial intelligence. So I am an engineer by background and a neuroscientist, and I really got interested in this area of understanding how the brain works because I have family members who suffer from mental illness. And so my interest has been in understanding the brain so we can ultimately come up with new therapies and diagnostic strategies to ultimately improve human health. And the approach that my research is based on is that the idea that, everyone talks about the idea of chemical imbalances in the context of psychiatric illness. The brain doesn't entirely distinguish between chemistry and electricity. In fact, electricity in the brain is chemistry. So it is the movement of ions, things we think about like sodium, potassium, and calcium. That is ultimately brain electricity. And so my approach was to try to understand electrical encoding in the brain, so how the brain uses electricity to encode emotions. And we approached this with the goal of understanding how about the 200 billion cells that we have in our brain, a little under half of them produce electrical pulses and respond to them. We wanted to see if we could understand all of those individual units of activity, how they work together to encode emotions. This starts off as an engineering problem because we've got to get in there and sample electrical activity. And so the project and work in my lab is largely based off of recording electrical activity using electrodes or wires, each the size of a piece of hair, from many brain sites simultaneously. And over time, the field has given us more and better tools to record more and more units of cellular activity. This has been a huge national thrust through efforts like the Brain Initiative to give us the ability to get more and more electrical information out of an animal's brain. Now, this produces an incredible challenge that you can appreciate. So I mentioned about 80 billion cells per animal. We're trying to understand patterns that happen across animals and ultimately scale into humans. And so it becomes a challenge making sense of that much data, right? And it was sort of a challenge that we weren't making great progress on until this idea, these tools based in machine learning or artificial intelligence suddenly gave us the ability to find patterns in very, very large data sets and relate them to behavior. So much of the incredible progress that we've made in terms of understanding brain activity and brain electricity has been through our ability in collaborating with folks across disciplines, particularly those in computer science and electrical engineering, and bringing these modern computational tools to taking large data sets and reducing them into smaller dimensions that we can then figure out how those small dimensions of brain activity interact with things like antidepressants or antipsychotics, how stress or drugs of abuse change those dynamics, and some of the newer work we're taking on in the area of thinking about psychedelics. So it's a tremendous honor and a pleasure to be here. I'm sure there'll be a lot of questions about how we're thinking about brain electricity and how we plan on ultimately developing tools that create therapeutics to change brain electricity. And I'm looking forward to spending some time with you all this afternoon. Thanks.
Great. Thank you so much. And, I think it's fascinating what you're talking about there, because a word that I continue to hear now in some aspects of neuroscience is the electrome, and trying to include these ideas of connecting electrical activity to all these other components. So that sounds great. Thank you so much. Okay, we are going to go next to Dr. Alberto Pareda. And while I believe some slides might be getting ready, I can tell you that he is a professor in the Dominic P. Purpura Department of Neuroscience at Albert Einstein College of Medicine, interested in the properties and dynamics of gap function electrical transmission in the vertebrate brain. And this includes investigating electrical transmission at auditory synapses, and as well as in fish, as well as in mammalian brain structures. So we've all been talking about plasticity for a long time in chemical synapses, and less is known about their modifiability of these electrical synapses. So I think we're going to hear some interesting discussion of that right now. Alberto, I will turn it over to you.
Oh, thank you very much. And thank you for the invitation. I'm looking forward to this discussion. As you said, I've been interested in synaptic transmission, but focusing on the learnings that model mixed synapses in fish had taught us during all these years, that I believe there have implications for the operation of circuits, right? And so I'm going to focus on the way I think that there is important, right? Our research fits into how the brain computes electrical and chemical signals. Can I operate my slides or from here, or? I think
You should have a button to control the screen on your side.
Where? I cannot use my keyboard, my keypad, my keyboard, sorry.
Yeah, there should be a button on your toolbar that should pop up. It should say like remote control operation. You can click on that. You should be able to click through the slides just with your mouse.
Where in the screen itself?
It should be like
Oh, and the clicking on the slide. Okay. Can I go back now, please? Okay. So this is the first slide. There are many ways that neurons communicate with each other, right? But there are two main forms of communication that happens within the time frame of decision making in sequence, right? And one is the chemical one, which is here on the left side. We recognize a complexity based on the forms of transmitter release and forms of receptors that different types have different type of consequences in their activation, and generates basically amplification of the presynaptic signal. Has been very well studied, and a lot of advantages from the evolutionary point of view. That way that we can modify the presynaptic signal into many other ways of configuration. And this the electrical one that we know much less, because we all try to oversimplify. And so there is a complexity we're recognizing chemical synapses, but there's still electrical synapses to be simplistically communicating the two cells, so the electrical signals can go through the other side by these channels called gap junctions. But there is a main difference, right? That actually has a very important functional consequence. Chemical transmission is episodic. The probability of release is less than 1, and depends on overwhelming majority of cases of an action potential. Well, here the probability of this synapse to be working is 1. It's always working, and they're bidirectional, and that generates what we call electrical coupling. Electrical coupling with both cells are electrically coupled. So always open, electrically coupled, leads and position of in different rather than in synaptic terminals with coupling between dendrites, somas, or axons, generates a diversity of functions, right, which are can excite or inhibit, synchronize or desynchronize, augment or diminish rhythms, introduce phase shifts, detect coincidence, and enhance signal relative to noise ratio. So it has another complexity of transmission that normally we don't recognize because we believe that chemical synapses are the epitome of synaptic transmission. So why this is important? Because we are trained to think about how the brain works in the circuits on the example of the law of dynamic polarization of the neurons, right? In which is formulated by Cajal. This is one of his drawings. And you see these little arrows where the information is coming, and that information comes through the synapse to the neurons, to the dendrites, and then it leaves through the axons to the other side, and finally is integrated by cell. So these properties of always open and bidirectional challenge this perspective. And that's the perspective I want to bring to the discussion, right? That chemical synapses, yes, that forms the circuit and the direction, but there is another form of transmission that is contributing to that based on bidirectionality. We have been studying not only the properties of electrical synapses, but the interaction with chemical synapses. And this is an example of, again, of the upper left, two forms of transmission, and they are both regulated by, for example, neuromodulators. It could be amines, dopamine, or it could be peptides, and both chemical and electrical synapses can be modulated and plastic. But our learnings from studying mixed synapses that combine both chemical, in this case glutamate, and gap junctions, electrical synapses, that taught us that actually these two forms of transmission interact with each other in a way that activation of receptors, in this case the receptors in the postsynaptic sites, goes and regulates and changes the gain of this electrical synapse. So there is a functional crosstalk, and this is not an oddity of fish synapses, because it was later found in mammals, both in the inferior olive and the retina, that nearby glutamate receptors from purely chemical synapses can, if they're close enough, to interact with electrical synapses located within the same postsynaptic cell. So there is a clear crosstalk, and in our experience, this is very short range, in which two synapses, chemical and electrical, interact with each other. And there is, in some way, also a new notion in that maybe we should think about the two synapses in the postsynaptic side be absolutely working together, part of the same structure. Further, I mean, we found even more complex interactions between chemical and electrical synapses and mixed synapses that I'm sure it will also show up in mammals, like activation of glutamate receptors, in the case mGluRs, lead to the release of cannabinoids that lead to the release of dopamine from the nearby varicosities, which in turn activate receptors and again go to regulate electrical synapses. So there is a very rich interaction between the two types of synapses that actually we have missed because we don't think about too much. Furthermore, the interaction doesn't stop in these modalities. It is a very well-known form of interactions in electrical and chemical synapses during development. The first synapses within circuits are electrical synapses that form blueprints, and they are later during development substituted by chemical synapses. In most cases, in many cases, they remain. And actually, this form of first electrical, then chemical synapses has been shown all through the animal phyla, from C. elegans all the way to cortex in mammals, right? So based on all those interactions between chemical and electrical synapses, also it's very obvious to think that, yes, they are also involved in pathological processes, right? So one of possibilities that we think is if you interrupt that interaction between the sequence of electrical versus chemical synapses, that could be lead to the dysfunction of formation of chemical synapses that has been shown in, for example, the leech and in the olfactory bulb and even in the thalamus. And also that this regulation by dopamine and receptors of these electrical synapses in the way that I showed it earlier today, they can go to, because they are expressed mainly electrical synapses in interneurons that generating gamma frequencies and will be in cognitive process to cognitive dysfunction. And also it has been shown that after injury, there's an increase in the formation of electrical synapses at the neuron that leads basically to increased toxicity and cell death. So I just want to finish with this idea that we all think that synaptic communication is electrical or chemical. I just hope that I can commission discussion that is electrical and chemical, right? And the information within neural secrets conveyed by combining those two forms that are equally complex in terms of contributions to the systems and complementary. And the molecular mechanisms that regulate this common expression and facilitate the function interactions is what we are investigating in the lab using zebrafish as an animal model. And of course, the individual properties and interactions add complex dynamics to circuits that, in my perspective, require the help of computer modeling and brings interesting computations that we can discuss later to our brain circuits that we should be aware of this interaction in the future. And thank you.
Thank you, Alberto. That was terrific. That has just added an unbelievable layer of complexity to something we already know is extremely complex. And a lot of people talk about how do we relate the electro with the chemical in the brain, and you're actually sort of dissecting that in really meaningful ways that I know we're going to want to talk about some more. I will mention, since I didn't mention at the beginning, is that as we go through these short sort of lightning talks, save all your questions because I think the Academy's folks and the board would like to have sort of a full discussion of what you hear. So each time you hear, think of a thought or a question as you're hearing someone speak, please jot it down because we will get to it during the discussion. So next, I'm going to go to Amina. I'm delighted to be able to introduce Amina, who has so many accolades to speak of. She is hailing from the University of Texas, San Antonio, where she is an associate professor, and she actually leads something called the Matrix Consortium, which is an artificial intelligence endeavor. She is also director of the Joint Graduate Group in Biomedical Engineering. She directs something called the Quan2 Project, which is a nationwide study to optimize brain health over the lifespan using and integrating biosensing technology, modeling, and functional neurogenesis bioassays. Amina, I noted that you were an NRSA fellow, which I also was when I was at NIDS many years ago, and yours was at the Johns Hopkins University School of Medicine. I will stop there because I want to give you more time to talk, but absolutely delighted to hear your comments today. Thank you.
So thank you, Diana. I saw I was muted. I'm a partnership lead for the Artificial Intelligence Matrix Consortium. It's started a few years back by Drisha Kuduthmpudi in AI. I want to share a little bit of context of following up on our two prior speakers in that looking at the chemical and electrical components of the brain, we also have an opportunity to tackle a larger medical problem, and that's what I have listed on the slide here. So there's over 100 million people in the US, children and adults, who have a neurological disorder, and that ranges from chronic migraines to strokes. That number is I would say small compared to the number of people who are affected. So if you think of doubling that number when you think of the caregivers and other people affected by the neurological disorders, you can see what a mammoth problem we're facing. But there's an opportunity, and that next statistic lists that opportunity, that 40% or more of neurological disorders associated with degeneration and some associated with development can be attributed to social environmental factors. So my research and many others have started to look at the components of what are the effects on social environmental factors and how can we start to measure it. Bringing in the computation here is a critical component. So one of the things my lab and others are doing is trying to measure in the environment what are things the brain is responding to. And you can think of right now, we're all in the room and we're responding to light, we're responding to sound, we're responding to social interactions, and our brain is synthesizing that information. If we exercised, we went out for a run, we came back, we also would be responding at the cellular level within our brain. But we right now don't have a clear way of quickly, in real time, analyzing our response to the environment. So a growth area in the field is to be able to do that. And that combines everything from imaging to being able to develop brain in dish assays, which are analog models of systems of the human that are responding to the environment. And so I want to share very briefly a little bit about what I'm doing in this area and what others are also tackling. And I know some of the other speakers here are also in this domain. I would just say that artificial intelligence falls into many different pockets here. So it can be covering everything from digital twins that span from what we're doing in the lab to what we can measure in the real world. It can also be computer vision algorithms to sense people's movement in the environment. It can also be algorithms that look at the biochemical interactions and the networks that are defined by the biology of the cells, from single cell sequencing of RNA, as an example, from RNA sequencing at the single cell level. So I just want to share that there's many components of where artificial intelligence and machine learning fall in. One of the areas that I'm focused in is understanding how we respond to the sensory stimulation in the environment. And when we think of this, many disorders are focused on understanding whether there's a molecular or cellular target. But a lot of disorders manifest themselves at the behavioral level in some ways that are common. And you can think of neurodevelopmental disorders like autism and ADHD and mild cognitive impairment and neurodegenerative disorders having some commonalities in how they affect regions of the brain that respond to sensory stimulation. And what I'm showing you here is an area of the brain that is commonly affected in many of these disorders. It's called the suprachiasmatic nucleus. It's a very small region, about 40,000 cells or so, deep in the hypothalamus that regulates response to environmental cues like light and response to exercise. It regulates our emotional responses. It regulates our response to a regulation of circadian rhythm throughout our bodies and throughout the brain. It regulates our temperature control, among others, and our blood pressure. It is entrained by the retina. So when we see light, as an example, the optic tract will be synchronized in ways that can stimulate the SCN, and the SCN in turn can stimulate other regions of the brain and define chronobiology throughout tissues of our body. So all of our cells that have nuclei have their own chronological clock. So they have proteins that are expressed at very specific times during the day that are regulated by this more global clock. And understanding that chronobiology can help us tackle many different problems. It can also help us improve how we develop and deliver drugs, as one example. My interest is studying this process, but it's very difficult to study it in humans. So it has been studied in many ways in other animal models. And to study it broadly, one of the goals has been to understand, does this area of the brain and other areas of the brain respond to sensory stimulation in a way that induces repair or changes? And there have been quite a number of studies in animal models of different forms, in fish, in rats and mice, in C. elegans, to look at how the brain changes specifically in response to sensory cues. And so during rest or sleep, there's been known attributes of brain changes, including microglial changes, dendritic pruning, gliogenesis or growth of new astrocytes, neurogenesis, and of course, electrical, protein, and cytoskeletal changes. But these have been yet to been shown in humans. It's very challenging to show what's happening in humans at the cellular level in response to environmental cues. And so one of the questions is, how can we start to tackle this problem? Can we develop methods to do it? And one of the ways people are doing it beyond my own lab is to look at imaging modalities that can get deeper and deeper into the brain and reach cellular level changes. But even with those modalities and that growth is a problem that we can't control or develop experiments in humans. No one will volunteer for that. So one of the aspects of my research and others' research is to be able to start to track what we can in humans and develop analog models in the lab. So what I mean by that is, as an example, we can start to characterize everyone in this room's basic biology of a neural signature. What I mean by that is we can measure proteins in your blood that are changing over the course of a day, that are changing in response to stimulation. We can measure how when you're going to sleep changes in response to what you're doing during the day. And we can do these by simple commercially available devices, like as a start or a ring or a Fitbit. And as an example, in one pilot study, we looked at the response of changing the amount people walked. So people walked 30% more steps than they had been walking in prior months. And we asked before and after they did that, did they have a change in their cognitive test scores? And did they have a change in when they went into specific parts of sleep and when they went into deep sleep or when they went into REM? And as the first pilot, we were seeing some patterns and when they went into rapid eye movement sleep as a function of whether they had an improvement in response to exercise in their cognitive test scores or whether they didn't improve. And so this is the first part of us being able to catalog more broadly individual signature responses to sensory stimulation. And that Fitbit is now has changed. It's broadly more behavior that we're capturing. So we have a collaboration with the Southwest Research Institute in San Antonio to capture using markerless motion capture people's activity and social interactions in the environment and their movement over time. We also have, of course, wearables as a part of this, but there are other measures. There's other new modalities. There's the electroretinogram you can record from the eye. There's EEGs that are wearable that you can have to be able to measure people's activity in real time. We're also using imaging of the eye to be able to see at the cellular level what's happening in humans. So those include corneoconfocal assays, so we can see in the cornea cellular changes, and they also include optical coherence tomography imaging of the eye or the retina. From these, we are characterizing people's baseline levels and how they change in response to sensory and environmental change over time. At the same time, me and many other labs, my lab and many others, are developing brain in the dish models of regions of the brain of interest. And for my lab, we focused on the suprachiasmatic nucleus model. This is, again, the area of the brain that regulates response to sensory stimulation and lies within the hypothalamus. And so what we've been doing is we've been differentiating induced pluripotent stem cells from blood into the hypothalamus and into the suprachiasmatic nucleus, and then recording from it in response to sensory changes. And so we can record electrically from multi-array recordings. We can record in collaboration with a researcher at Baylor who's developed voltage sensors, Francois Saint-Pierre. Voltage, I'm showing you some calcium imaging as well. At the same time, we can look at molecular and cellular changes in these cells. And again, these are models of the individuals that we can study in the environment. Some of them are. They're developed and induced from the blood of the individuals we can study. Some of them. I would say there's a challenge here in that every time we develop one of these assays, it can be over $5,000 to develop one of these cell lines and many, many months. So 6 months or more to well characterize them into the stem cell stage. And that's beyond them developing into the brain models we're interested in. But this allows us an analog of a region of the brain which we can't easily access in the human. And many other labs are focused on other regions of the brain to be able to model function, not only in recording in these cells, but also stimulation. Can we model the response of these assays to sensory things that would be similar, analogous stimulation in the real world? So different changes of light spectra or different levels of oxygen or carbon dioxide in the environment. Even pollutants you can think of exposing these cell models to. So computational analyses is the focus of what we're doing for what we're discussing on this meeting is a big part of what we're doing. We couldn't do this work without using computation. And I'm just showing you one example of us tracking people's behavior before and during the pandemic in response to a stimulation, which we didn't, of course, predict, and that is the social constraints of having stay-at-home orders, which changes people's activity, which change people's social interactions. And we were just characterizing attributes of when they went into REM, and over time, what other aspects of their day-to-day activity changed in response to these environmental cues. This is one example of what you can characterize for an individual. There's, of course, many more. But what you can do computationally is track for every individual, what is their trajectory over time compared to their own. So meaning that everyone is their own control over time. You have a baseline of their behavior. And so here you can see that we used unsupervised learning to be able to predict patterns, be able to classify patterns of people's activity and identify those that improved after the pandemic or those that stayed the same or worsened their activity during the pandemic and afterwards. Another aspect of computation that comes in here is, of course, the molecular analyses of the cellular models. So we do high throughput screening of the cell lines, and that includes looking at many different proteins at once in response to chemical cues like light or a compound of interest. And in that sense, we're using computation in two main ways. One is the molecular readout of these cells in a dish assays, and the other is using computer vision to characterize their changes over time as they're responding to the sensory stimulation. And then I mentioned tracking. So tracking is a big part of what we do. So our collaborators at SWRI has developed motionless, markerless capture methods, which we're now implementing in city parks in San Antonio to capture people's changes as they're walking around. But the computer vision algorithms also are useful for tracking cells in a dish in 2 and 3D, and networks in a dish. How do they respond as a collective in response to sensory and environmental stimulation? And we're among many others who are developing computer algorithms that help in neuroscience. So I wanted to share that. The other thing that can be done from an artificial intelligence point of view is develop digital twins, where you try to match what you can actually measure, the same modalities, whether they're chemical or biochemical, whether they're electrical or whether they're physiological. Can you match or develop an analog, a transfer between what we can measure in a dish to what the human model or human measurements can give us? And for this, we're developing parallel models or developing a digital twin where we can ask the question of, if we see this electrical signal in a dish and it's responding to a sensory stimulation in the environment, is there an analog? Can we predict that same response in the human level at a different scale? And computer modeling and first pass models are enabling us to develop the framework for this. So I'd like to just stop there and just share that there's a lot of collaborations across many different domains, artificial intelligence being one of them, but neuroscience, molecular biology, and other domains, are chemical engineering and synthetic biology are all a part of what we're doing. Thank you, and I look forward to the discussion.
Wow, thank you, Amina. I have so many questions right now, but I'm gonna hold them. But I will say, I find this really exciting. I've a lot of interest in the modeling you're doing. I love this idea of neurosignatures and how you're trying to. We talk a lot about wearables and what we think wearables can do, but this idea of a neurosignature, you're getting at really what's behind that. And what connects what the wearable is telling you versus the underlying neural activity there. I think that's fascinating. Also, I think being able to study kids who stayed home during COVID-19, because we've been hearing a lot about the impacts of that, potentially negative impacts on that of children's development. And so you're actually developing a way that we could look more scientifically at what right now I believe is kind of anecdotal, and that would really educate future public health approaches. So wow, fascinating stuff, lots of questions, but we have to move on. We're going to, I apologize, I'm going slightly out of the order here because I have two different lists of folks, but we're going to get to everybody. We're going to go to Sapita next and then Carrie, and I'm saving Gabe for last, because that's on a slightly different topic, which is on the ELSI issues, and I want to make sure we spend the appropriate time on that. So let's go to Sapita Sadagiani, who is Associate Professor of Psychology in the Cognitive Neuroscience Program and Bioengineering and faculty of the Neuroscience Graduate Program at the University of Illinois Urbana-Champaign. She directs the Connect Lab at the Beckman Institute for Advanced Science and Technology, where she is full-time faculty. Her PhD in neurobehavioral and sciences was from the Max Planck graduate school, fantastic, and has also done training at Berkeley and Stanford. Also, some connections to NIMH and NINDS. So let me go ahead and turn it over to Sapita.
Thank you so much for having me here. It's a pleasure to listen to the very diverse set of content. Let me just make sure I can advance the slides. Okay, this seems to be working, except I clicked on Zoom of some sorts. Let's see how I can do this. All right, I'm sorry about that. I was asked to talk about neurocognitive networks and functional connectivity in the context of cognition. And when we talk about neurocognitive networks, we typically mean networks at the largest scale of the brain. So we look at the brain as a whole system and look at remote regions that exchange information to support cognition. If we keep the spatial scale at this largest scale, I want to focus on time scales of connectivity, which is, I think, very underrepresented in this large-scale connectomics field, which is very fMRI focused. So my focus for today is talking about the scales in the time domain and why that's important. So just some terminology to begin with. Neurocognitive networks, also called intrinsic connectivity networks, or perhaps somewhat misleadingly resting state networks, again, are these networks of regions across the brain. And they are intrinsic because they are observed irrespective of the mental state that we're in. And so intrinsic is anything that is not evoked in response to direct response to external stimuli or events or cognitive demands. So neurons are continuously active and their activity fluctuates over time. And in fact, most of brain activity is intrinsic and is not evoked. This spontaneous activity is highly temporally aligned between certain sets of brain regions, while it's less related across other sets of brain regions. So if we generate measures of temporal concordance over the recording, typically in fMRI, between all possible pairs of brain regions for a certain brain parcellation of choice, we get this connectivity matrix, which is termed the functional connectome. The functional connectome at this large scale is characterized by sets of regions of particularly high connectivity. Are you able to see my mouse moving when I'm pointing, or? Okay, great. And so these networks, we have actually showing spontaneous co-fluctuations of activity. This is in total absence of any motor imaginary movement or even physical movement. But then it took about a decade to really realize and actually accept in the field that there was a lot of pushback at the beginning, but that these neurocognitive networks are not restricted to primary auditory, for example, here, or visual, but they actually include higher order networks, such as the dorsal attention network here, and in fact, all of the large-scale neurocognitive networks that we have known, again, previously from task settings. So because most of the this architecture is intrinsic and ongoing irrespective of active mentation, early on, Marcus Raichle hypothesized that this is really going to lead to a paradigm shift in neuroscience, and that we will start understanding the brain as an inside-out predictive organ as opposed to an outside-in reactive organ. And that has, at least in the large-scale field of cognitive neuroscience, turned out to be true. So fMRI has also shown that when we look at the connectivity matrix in a time-resolved manner, we actually see time-varying dynamics. And in fact, those dynamics within individuals can predict perception into the future. So if we apply machine learning to fMRI data, prior to actually giving the stimulus to the participant, we can predict above chance what the participant is going to respond before the stimulus has even happened. So here's one example. This is the discs represent brain regions. The colors are the neurocognitive networks that we looked at. This is not anatomical space, but is a space that emphasizes the topology of the whole brain network. So we gave the participant faint sounds, and on some trials, the participant heard them. Sometimes they were missed, and you see much higher segregation of these neurocognitive networks, much more modular structure. And again, this leads us to be able to predict perception ahead of time. Across individuals, also, these dynamics, as observed from fMRI, are predictive of cognitive abilities or associated with cognitive abilities. They're associated with psychiatric outcomes, with neurological outcomes, even predicting into the future, at least for very severe cases of neurological disorders. They are brain traits because they're highly individually specific and genetically determined. But there is a problem, and that is that almost the entire large-scale connectomics field is using functional MRI. And when we think about cognition, it doesn't really happen at the speeds that MRI is particularly sensitive to. So fMRI measures dynamics in the range of several seconds. But if you look at cognitive processes, you'll see a range of time scales. So this is just an example. This is a piece of a nursery rhyme, and this is the air pressure wave. If you look at the amplitude modulation of the phonemes at a pretty fast speed, the amplitude modulation of the syllables is a little bit slower. Prosodic stress points, there's a melody to this. It's a little bit slower, and then all of this is embedded within sentence level semantics and syntax. And so there's a hierarchy of timescales that matter for cognition. And each one of these processes is actually requiring interaction between distributed sets of brain regions. So networks are required at all of these different speeds. And fMRI just sees a sliver of this. And so what we've done in our lab is actually moving to non-invasive scalp EEG. And I'm not going to go into the details of the methods unless there's interest, but I put this figure up here because I want to emphasize that most of the cognitive neuroscience community is still heavily underusing EEG or even MEG for the study of large-scale connectivity. And the reason, presumably, is the worry that source leakage, which is when a single source of signal in the brain projects to multiple sensors and artificially causes the appearance of connectivity, is really a big concern. But there's algorithms to correct for source leakage to some degree. It reduces the signal by a whole lot, but still leaves a good amount in there. So we generate these connectomes in source space through source space projection and then leakage correction. And even in quite large cohorts, we have shown that what is obtained in terms of very, very rapid sub-second dynamics using EEG, types of dynamics that are not visible in fMRI, we see high variability, individual specificity that is associated with cognitive control abilities. And these are healthy young adults. So I think our work and other work really suggests that we can trust this data modality to a sufficient degree to give us access to whole brain connectome dynamics at a rapid scale that matters for cognition. So this is my last data slide. What I'm particularly excited about is putting the data modalities together. And so we've been recording for many years now concurrent scalp EEG with fMRI, and we have graciously gotten access to very rare data of intracranial invasive EEG in humans with concurrent fMRI from the University College of London. And both types of data modalities, invasive EEG and the lack of source leakage, as well as the corrected source reconstructed scalp EEG with fMRI, paints an interesting picture when we look at what happens. So both data modalities are recorded concurrently. We project them onto the same brain parcellation. We look at time-resolved connectomes over the recording. And so I'm showing here about 2.5 minutes worth of recording from one participant as a scalp EEG with fMRI. fMRI is up here on the top, and then there's canonical frequency EEG here. The color code shows recurrent connectome states. So without going into too much detail, the connectome states here were defined as all possible combinations of co-activation among these canonical neurocognitive networks. So let's say a pattern where the default mode network is co-activated with the visual network would be one connectome state. If the visual network is co-activating with somatomotor, this would be another connectome state. And we have observed that all of those combinations actually occur in the brain at all of these different speeds. But what you can even see just visually is that there is at least no linear relationship between what's happening at the faster speeds and the slower speeds. Simply convolving neural activity at the fast speed with the hemodynamic response function does not give you what fMRI is observing. It seems that they are tapping into different speeds of the connectome. Because the states are spatially related, they all incorporate these neurocognitive networks to some degree. We can imagine the total connectome dynamic state space as one space across these different time scales. And imagine that fMRI is really good at measuring the very slow dynamics of the connectome, and it's moving along this trajectory where the connectivity patterns ever so slightly change bit by bit. But at the same time, there is other trajectories across different parts of the state space covered by faster dynamics that are concurrent. And so what's interesting about this model is that if you pick out one single moment in physical time, let's call it T10, right? We don't have one connectome state, which is what the fMRI-centric view has been telling us for over a decade. We actually may have something like a stack of connectome states. That means a certain set of brain regions might be highly co-fluctuating, timing their sending and receiving of information in this kind of a network pattern. But there might be another set of regions that is highly connected at a different speed of neural activity. And this is all happening concurrently, which could provide a model for having different independent sets of networks processing information in order to essentially provide a basis for the different speeds of cognitive processes. So just to summarize, the dynamics of the connectome comprise multiple parallel streams if we actually move to a multimodal setting. They have similar spatial patterns, at least part of the variance is shared in terms of the spatial patterns that occur. And the statistical probabilities that dictate what pattern follows the next are also repeated across the different time scales. So there are principles that are scale invariant, which is really interesting from a complex systems perspective. So the sequences look similar, but they're stretched or squished in time, depending on what neural timescale we're looking at. So we have different speeds of these parallel streams, which I think really speaks for studying the large-scale connectome using a multimodal approach, because it is not simply possible to have the two data modalities separately and think that you can convert one into the other, and you will understand how it relates to cognition. So this was my final conclusion. Thank you.
Wow, thank you so much. That was fascinating. I'm in my mind now, I'm thinking of a whole new definition of that famous book title, Thinking Fast and Slow. There's a whole other level of complexity there that you revealed that is absolutely fascinating. And I think we're going to have a lot of questions about that. So we have two more speakers. I'm going to go to Carrie Johnson next. And as I mentioned, we're saving Gabe for the end. But to introduce Carrie, she is a program director at the National Institutes of Neurological Disorders and Stroke, a job that I once had and enjoyed very much. She's been working in her portfolio on brain connectivity across scales. Its acronym is CONNECT, projects aimed at large scale mapping of local and long range neural connectivity. But prior to that, she was at USIS in Bethesda, also another great place where she studied basal ganglia circuits in reinforcement learning and behavioral flexibility. And she hails originally from Vanderbilt and did some work at NIAAA. So Carrie, delighted to have you from my old stomping grounds at NINDS.
Thanks for inviting me to participate in this panel today. Yeah, I'd like to talk about the Brain Connect program in the context of larger work that the NIH Brain Initiative is doing, and I'll highlight some areas of impact from the Connect program that I think are particularly relevant to the focus of today's discussion. So for those of you who aren't familiar with the Brain Research through Advancing Innovative Neurotechnologies, or Brain Initiative, the mission is to revolutionize our understanding of the human brain by accelerating the development and application of innovative technologies. The Brain Initiative was announced by the Obama administration in 2013, and the first awards were made in 2014, so we're celebrating our 10-year anniversary this year. The Brain Initiative is a partnership between 5 federal agencies as well as several private foundations. The central, oh, sorry, I'm not used to this control here. The central goal of the NIH component of the Brain Initiative is to develop and apply new tools for understanding how neural circuits underlie complex behaviors in health and disease. And the priority areas are guided by 2 strategic reports, the Brain 2025 and Brain 2.0 reports, which you can find on the NIH Brain Initiative website using the QR code on this slide. This is an effort that spans 10 neuroscience-related institutes and centers at NIH. Along with the focus on technology development, we support work that leverages emergent technologies to enable new discoveries about circuit function that provide a critical foundation for discovering new therapeutic strategies for human brain disorders. And to maximize the impact of these efforts, there's a major focus on disseminating and democratizing technologies for both basic neuroscience research and clinical applications. Based on the recommendations in the Brain 2.0 strategic plan, the Brain Initiative has launched 3 large-scale projects that promise to transform neuroscience research, illuminating foundational principles governing the circuit basis of behavior and informing new approaches to the treatment of human brain disorders. The Brain Initiative Cell Atlas Network, or BICAN, builds on findings from the Brain Initiative Cell Census Network, its predecessor, which was a collaboration of over 250 NIH-supported scientists at nearly 50 institutions across 3 continents. These projects enabled major advances in understanding of the location and function of cell types at the whole brain scale, facilitated comparisons across species, and scaled up technologies used to acquire data at the single-cell level. Now that researchers have made such tremendous progress identifying the types of cells in the brain, a critical next step is to understand how they are all connected. The Connects program emerged based on recognition that understanding the human brain requires a really ambitious scaling of current technologies to generate connectivity maps of whole mammalian nerve systems. This will involve an interdisciplinary effort from fields including neuroscience, engineering, computational and molecular biology, mathematics, and computer and data sciences. The tools, discoveries, and resources that emerge from this project have the potential to transform how neuroscience research is conducted in much the same way sequencing the human genome transformed how biomedical research is conducted. In parallel, the Armamentarium for Precision Brain Cell Access will leverage new brain cell census data and advanced technology to build an expanded toolkit for monitoring and manipulating specific brain cells and neural circuits in model systems that include both laboratory animals and human tissue specimens. The goal of the Connects program is to scale up technology and research capacity such that whole brain mapping and reconstruction of comprehensive wiring diagrams will become feasible. This includes development of infrastructure and tools to disseminate data and integrate data across modalities, and the resulting brain-wide connectivity maps will provide conceptual foundations for new discoveries of neural circuit function. To achieve these objectives, the first 5-year phase of the Connects program is supporting a variety of approaches that are aimed at scaling up to entire brains, and I'll just quickly go over those in a minute. And projects are acquiring data from large subvolumes of the brain to demonstrate feasibility of future scaling. These efforts will generate proof of concept data sets to understand wiring principles of specific circuits and their functional implications, and will allow us to realistically assess the potential to scale up to brain-wide connectivity mapping in the near future. The Connects program launched this past September and currently supports 11 grants projected to total $150 million over the next 5 years. These awards support diverse research teams and their collaborators across 40 universities and research institutions from around the world. In this first phase of the program, the projects are spread across 3 main types of approaches. The first looks at electron microscopy to map the mouse brain at synaptic resolution. These projects are improving sample preparation, increasing imaging throughput, building scalable data processing pipelines and analytic techniques, and reducing the time and costs associated with imaging and analyzing large volumes. The second theme will develop nucleotide barcode sequencing approaches that label individual neurons with unique barcodes and map their brain-wide projections or inputs. This innovative approach represents a tremendous increase in throughput for single neuron tracing and is also compatible with single-cell or spatial transcriptomic techniques, so this allows integrated determination of cell type and connectivity information. And the third theme includes projects that are using novel multimodal imaging methods in the brains of humans and NHPs, larger brains, to understand the topography of long-range projections across different scales and resolutions. These projects combine a variety of imaging approaches. These include diffusion MRI, polarization-sensitive optical coherence tomography, and various other high-resolution optical techniques. And this work has the potential not only for increasing our understanding of brain connectivity in large brains, but for having immediate clinical impact in terms of potentially optimizing targeting of deep brain stimulation for various neurological and psychiatric disorders. I'll just briefly say that the efforts underway in the Connects portfolio have a variety of implications for understanding brain functions at scale. Of course, having more detailed connectivity information will allow generation of new testable hypotheses about structure-function relationships and will provide new constraints for computational models of circuit function. New computational approaches for automated reconstruction of neurons and circuits, integration of data across modalities, and AI enhancements of data acquisition will be applicable to many other research problems. As I mentioned earlier, projects that are studying the topography of fiber tracts across long ranges at very high resolution have the potential for immediate impact on clinical practice as well. There's also a heavy emphasis on dissemination of technology and development of knowledge bases to make sure that these efforts are broadly useful to the neuroscience community. And finally, these types of projects inherently foster interdisciplinary team science and bring together people with diverse expertise and perspectives, which is critical for successfully tackling the biggest problems in neuroscience. I'll stop there, but if you'd like to learn more about the innovative work being supported by the NIH Brain Initiative, and for anyone interested in exploring funding opportunities, you can use this QR code to visit braininitiative.nih.gov. Thank you.
Carrie, thank you so much. That is awesome. I love to see the follow-on to the Brain Initiative with those goals and milestones attached. I think there's tons of opportunity there. So thank you for that. I'm sure folks are going to have some questions about it. So we're going to go to our final speaker, who is going to talk about some of the ethical implications of all of this work, and that is Dr. Gabriel Lizarra Munoz, who is Assistant Professor of Psychiatry and Global Health and Social Medicine in the Center of Bioethics in the Department of Global Health and Social Medicine at Harvard Medical School, as well the Department of Psychiatry at Massachusetts General Hospital. His background in neuroscience, law, and bioethics is about examining implications of emerging biomedical technologies in neuroscience and genomics. He's also been funded by the Brain Initiative that was just mentioned by Carrie at NIH, NIMH, and the Human Genome Research Institute. So thank you, Gabe, for patiently waiting, and we'd love to hear your presentation now.
Great, thank you for having me, and thank you for that introduction and all these wonderful presentations. So I'm going to start with talking about informed consent. Everyone knows something about informed consent. So basically, where I want to get to this is that the integration. So even though we're learning a lot about how to integrate all these technologies and generate novel information that will hopefully help us understand, yes, as you know, others were saying before, understand different health conditions, different neurological conditions, etc. The more complex these studies are, the more complex the informed consent process becomes, right? And so I just want to bring up informed consent, you know, as an important aspect of this, because it can sometimes be, you know, overlooked, and I think it's really important that we understand that a lot of the information, once you start integrating data sets, a lot of the information that you're going to generate are things that, you know, as my background is, my PhD was in neuroscience, and so there's a lot of things that we don't know what to expect once we start integrating a lot of these data sets. So it can be quite complex as researchers to know that, but also to convey that information to participants, and it's really important that we work on developing that trust with participants, that they understand what kind of information we're going to be generating, and how we're going to be combining that information. There are going to be limitations, of course. You know, there's even things that you know may not know how, you know, what's gonna come out of the way that you integrate the data. But we really need to make sure that as we integrate all these data streams, that participants also have an understanding, and not just, and I say, like, you know, promote understanding, not just disclosure, because our system tends to be very disclosure based. And but you know, at the end of the day, if something happens where a participant feels like they didn't, you know, understand what was being explained, etc., in the informed consent process, you know, it's usually about understanding, not about the disclosure component, right? So you know, when you look at the informed consent forms, like, they'll have pretty much everything possible, right? So I think it's really important that we as we invest in developing the systems that allow us to better understand the brain and to combine data sets, that we also invest in ways to explain what we're doing for participants, and communication tools that will allow them to understand the way that these data sets can potentially be integrated. Of course, there's limits to this, but I think it's really important, as this gets more complex, that we also kind of raise the bar on the you know, expectations that we have about consent, so that participants can be partners, and you know, as much as possible, and understand what's going on. Another aspect that I wanted to bring up, and this is in part coming from the sort of LC, ethical, legal, and social implications of genomics world. There's been a lot of debate in genomics about, you know, how to manage clinically relevant findings. So let's say you conduct whole exome sequencing, whole genome, etc. You know, you may generate incidental findings, you may generate, you know, secondary findings, things that you that, you know, maybe were not part of the primary you know, data that you were looking for, but it came out in the data, and how do you manage that information? And I think that's something very important in this context as well, that you know, as we continue integrating data sets, and we've heard from a number of different presentations, and at the end of the day, like, you know, that's a lot of the what we're trying to achieve, is to help generate clinically relevant information that will help promote health and well-being of many patient populations. So it's really important that that we have some kind of process that you know, helps us identify if there's clinically relevant information that we're generating, and that we have a process for you know, one, figuring out how we're going to manage that data, which even in genomics can be quite complicated. Just the logistics of keeping track of the participants and where, how to communicate with the participant if something is generated. Similarly, like you will be facing a lot of those challenges, and perhaps even more challenging in a space like this where you're integrating potentially data sets from different groups, different parts of the world, and even the consent process may have been most likely was different. So it's really important that we start thinking about how are we going to manage that potentially clinically relevant information. Ideally, we want to maximize the investment that we're doing in the research, and so ideally, if we generate something that's clinically relevant, we would be able to communicate that information to the participant if it's particularly if it's something actionable, something that the participant could possibly do something about. That's a very important aspect, I think, of this. Related to it, one of the challenges, one of the logistical challenges, not only keeping track of the patients and, how to contact them, but also, many of the researchers that do this kind of work are not necessarily clinicians. So, how do you figure out, how to identify some of these clinically relevant findings if you're actually coming across them? Let me see if I have anything here. Yeah, and then another thing, related to that would be what kind of information the patient actually wants. It may be that, for one reason or another, patients actually do not want you to return any kind of information, and so we also have to have a discussion about how to manage patient preferences in a context like this where we may generate this kind of information. Related to generating clinically relevant information, but also, as some of the speakers were mentioning, there are, different sort of behavioral traits, psychiatric traits that you may be able to generate some information. And, as we continue developing these tools, we'll be able to do more of that. One of the challenges is that, in the genomics world, we have GINA, the Genetic Information Non-Discrimination Act. It's limited in its scope, right? It only covers discrimination or protects against discrimination in employment and in health insurance. But for this kind of neuroscience data and the integration of neuroscience with other types of data that has been mentioned before, there isn't any kind of law that protects specific biomarkers other than GINA for genomics. So one of the challenges is if this information gets out, the participants would not really have a lot of recourses to against if they face some kind of discrimination based on some of these findings. So that's another aspect that I think we should continue thinking about and looking for ways to help protect participants, help promote participation in this research, which was the kind of the original intent of GINA, was to protect people against discrimination, but also help them feel more comfortable participating in research trials so we could make more progress in genomics. And then, as we continue integrating all these data sets, we'll have the issue of how to protect the privacy of the individuals while promoting accurate data integration. And so the NIH and some other groups have been interested in promoting the use of global unique identifiers, which in many ways, could be very helpful to allow researchers to know, if a participant has participated in some other trial, and potentially integrating data from other studies. But that also comes with the risk of potentially re-identifying, and I know that what's being proposed right now has protections that are pretty strong protections nowadays, but we know how and I think we have to also take into account that I think most people, most participants don't really trust that their data can be protected, because we see so many instances of hacking of different kinds that it's it can be difficult to make the case that, hey, we'll be able to protect your data. So there's a consent component there as well, where we need to work to help the participants understand that their privacy can be protected to some extent, but there will be some limitations. And again, how do we protect individuals if this information it becomes it's hacked, etc. I think it's something that we need to continue thinking about, potential regulations or potential policy solutions for that. And I'm happy to discuss any other issues or questions that you may have, and thank you.
Gabe, thank you so much. That was excellent, an excellent summary. So let's, as I mentioned, I have a lot of questions. Kavita has noted to raise your electronic hands or place your questions or comments in the chat, which I can call on people right away as soon as I see them. If there are, I know there's a group of people in the room at the Academy. So if you're physically putting your hand up in your room and I don't see it, throw something in the chat, or someone else can flag it to me. So I think, we have a number of questions. So if I don't see any hands right away, so I'll take the moderator's privilege to ask some questions. I think we heard a lot about advances that are happening here. And I had one specific thought that was in my mind, kind of connecting some of the pieces of the different things I've heard. And I'll sort of I'll couch my question as an example scenario. So we talk a lot, particularly in the DoD, about wearables and all the cool things that wearables can do, and the ability of wearables to inform about military individuals' ability to perform, and their situational awareness, and what's happening with them when they're flying jets or doing other kinds of activities. And so one of the questions I had was, we heard today about the sort of electrical and chemical nature of synapses and synaptic connections. Perhaps a layer above that, we heard about the connectome and these sort of fascinating timescales throughout the brain that are happening at once, perhaps the next level up above these electrical and chemical connections. And then, Amina had some interesting discussions about wearables and trying to connect those to behavior. So my question is, if someone has a wearable, what actually kinds of things could we be measuring that give us meaningful information on that level? Are we just measuring electrical things? Are we measuring other aspects of physiology that give us a better window? Or are we measuring the connectome? What actually, where could we go with that? And I see a couple of hands, but I'm assuming Jerry and Nathan have questions. So I'll go to Amina, who might want to answer my question.
Thank you, Diane. So in terms of existing commercial wearables, there are trackers for sleep, for activity, for oxygen levels, for newer ones that are in development, research grade. There are wearables for EEGs that you can think of, that, and there are some that are now clinically being used, so people can wear them at home and record from them over many days. There are others that are emerging where you could record other attributes, like even metabolites in the brain, non-invasively through wearable devices or patches. So I would say that that area is an area of extreme growth. So there's going to be modalities to measure everything from molecular changes to whole brain electrical readouts. And adding to that behavior, so from the environment, can we understand what people's movements and social interactions are? Their voice is going to be recorded. So in terms of a neural signature, the depth of it is just expanding for wearables, for, I would say, not just wearables, but anything that can be passively measured in the environment that relates to somebody's cognition and their behavior.
Wow. And so if I may, and I think maybe Septida has a comment on this as well, and I'll let her speak. So we could conceive of a neural signature getting consistently more attuned to an individual, like a fingerprint maybe, but we're calling it a neural signature. I don't know if that's the case or not. But Septida, do you want to just chime in on that? And then I'll start going to the questions. We'll go to Jerry and then Nathan.
Yeah, I just wanted to continue where Amina mentioned the EEG wearables. And I mean, this has been around for a long time, the idea that EEG would allow to monitor brain states, including arousal and high stakes situations, right? You mentioned pilots and similar situations. So to look at the connectome and connectivity, that hasn't been done to my knowledge with any wearables for the reason that you need a larger number of channels and you need good data quality. So these wearables, especially the commercially available ones, they often take the forehead where the hairs don't disturb, or if they go in, there is low coverage. There's usually no conductive material involved. Saline solution is an easy one where you don't need the gel. So we're testing some of those out now. The technology is improving, but connectome dynamics as a online monitoring approach hasn't been tapped yet, as far as I know. And I think one of the reasons is I think generally EEG is underappreciated. What has been around for a long time is the power spectrum of the EEG. So you can look overall in the sensor space without source localization, even with few sensors, at the ratio of the different frequencies occurring. And they do track your attentional states, and they do track very well sleep stages. In fact, sleep is defined very much by the states of the EEG. I mean, that is how it is literally defined clinically. And so that has a lot of information. So much simpler EEG measures have a lot of information that can be tapped in wearable data, as long as the sensors have good contact. A lot of the commercial ones don't, which concerns me a little bit when people think that they can track their sleep and whatever, and the actual contact isn't very good, and the software doesn't make that very apparent because it's probably not in the interest of those who sell it. But I think a lot of advances being made there, and I have hopes that we can look at connectivity in terms of an online monitoring aspect, which I think it tells us untapped aspects of the electrophysiology that just looking at the power spectrum won't tell us.
Yeah. I couldn't agree more on that, on your statement about the EEG. And we have talked about this before. Kavita and I were on a different group committee where we had people present on some of these issues. And I agree. I think there's a wealth of information and development there that's going to be fascinating as this field progresses. So let's go to Jerry and then Nathan and then Mauro. I'm doing them in the order that I've seen them appear. So if I don't catch you, let me know.
Great, thanks. Is this like I want to thank the speakers. It's just an incredible array of talks. They're just fascinating, and echoing Diane's remark about the challenge in going from the molecular and cellular to the network to the macro. It's just unbelievable opportunities, but also an unbelievable challenge. I want to really go back to the electrical couplings, because I at least had never heard of them before. And I imagine they're somewhat new to neuroscience. And I'm reminded of, I have a 1911 set of the Encyclopedia Britannica in my attic. And every once in a while, I like to go to it and look up things like, how does the sun work? Because they had no idea what nuclear fusion was in 1911. And it's funny watching them sort of blunder around and trying to explain it without knowing that. And I'm wondering if now we know there's such a thing as electrical coupling, are there things that just did not make sense in neuroscience? We can go back and go, oh, there's something else going on. Maybe this unresolvable puzzle now is going to start to make sense.
Well, I can answer, or at least extend the discussion on that. Yeah, I mean, the discussion over the nature of synaptic communication was a frame by the idea of the circuits as imaging by Cajal, right? That there was a flow of information. And there was a discussion if the transmission was electrical or chemical. Electrical, not in the case of those structures, the gap junction general coupling, but fields effects that can excite the postsynaptic cell. As always, the discovery of one type of transmission, the chemical transmission, led to, as always, to think all synaptic communication is chemical, right? But that then led to the discovery that electric communication could be electrical by virtue of those electrical structures called gap junctions. We know they're all in all nervous systems, they both coexist, right? But for many, many years, we thought the mammalian system was more evolved, and if things like less relevant to the mammalian brain. If you look in your textbooks now of neuroscience, you have like a 1 page of electrical synapses, very poorly developed, and 3 chapters on chemical synapses. And they bluntly say in the Candel that they are less relevant, despite that we know, for example, structures on the CNS, like the retina, since the 70s and 80s, that they both combine, and how the combination that the both structures working together, they're important for all this signal processing that the retina does. So it's more about we breaking up with that notion, right? And bringing prejudices, right? To allowing to understand about electrical synapses in the brain. A big breakthrough was the cloning of the gap junction protein that forms the electrical synapses in 1998. That it was called the connexin 36. That is very interesting. There are 21 genes of gap junction proteins, but only a few are in the brain, and 99% of the synapses are formed by only one that like to form channels with itself, and it's neuron specific, right? So when you look at the message in the brain, it's everywhere. Every single structure in the brain is there. And it's a hallmark of inhibitory interneurons. All the inhibitory interneurons have been studied at electrically coupled. Cortex, thalamus, spinal cord, you name it. And interneurons, they're involved in generating the pulse for the oscillations and the activity of the circuits because of the inhibitory property. So for sure, they're contributing. But we have to start thinking about when it comes to how they do operate with the circuits. That's where the challenge comes, right? So it's hard because they do things that chemical synapses do not do, right? And it's very hard to understand the relevance because of that. But necessarily we have to include them. One of the things we do in the lab is to study the plasticity of electrical synapses. That we came up with that concept, which people thought we were crazy. That's an oxymoron. There's just channels that they're not plastic. They don't change. So we show that by interacting with chemical synapses, they undergo plasticity. And we are trying to understand which is the molecular organization of the electrical synapses. Where's an electrical synapses beyond those channels, right? And we are finding that it's not as different in many aspects of chemical synapses in terms of molecular complexity. They are just different, right? They're just different. And at some level of the secret, I agree with you, we're missing something, right? It's like the dark matter of the brain is still there. We don't know, right? So, but that's the challenge. And I think that's why we need computers and models, right? Because when you put 2 or 3 things together of that nature, right? Like electrical synapses, they do also of operations I mentioned, but the most coincidence detection, for example, that they can detect fuse enough because they're coupled together. They can detect if they both receive at the same time, right? The signal or not. All those operations are done beautifully by electrical synapses.
Thank you.
Thanks, Alberto. And I think you articulated one of the questions on our agenda are what are some areas for innovation and future work in this space? And I think you've just articulated very clearly some of that. I think that Kath may also want to respond to Jerry, and then we'll go to Nathan.
Yeah, I wanted to follow up on that. It's an interesting example that you talked about the sun and our lack of understanding of nuclear fission. So it turns out the Nobel Prize for Physiology and Medicine in 1906 was shared between Cahal and Golgi, who really didn't get along and had a lot of different ideas about the brain. And some of those ideas was based on the size that there was individual cells and units of sort of computation, and another hypothesis in which there was like this large interconnected web of brain cells. And it turns out, as Alberto mentioned, this idea that, particularly through the inhibitory system, these gap junctions are connecting the inhibitory neurons electrically. It sort of does support this idea that there's this interconnected web of cells directly transferring information from one to another. It's just something that, neuroscience as a whole hasn't thought a lot about. I barely heard about gap junctions during my training as a graduate student in neurobiology. On the other hand, I heard about gap junctions all of the time in medical school, right? And so the heart, like the brain, has a fast conduction system in which cells release neurotransmitters and change the function of their target cells. But the heart cardiomyocytes actually have gap junctions between them that transfer electrical activity from one cell to another. It's actually how the heart helps synchronize itself and why you get large synchronous beating of large amounts of tissue. And so it's been appreciated that gap junctions are electrically active, are in the body, are in tissues throughout the body, 21 isoforms. We got really interested in this question of gap junctions a few years ago because we were generating these electrical maps of fast brain activity and how they related to emotional states. And we really ran into a challenge thinking about, well, if you create a diagnostic framework where you're essentially finding patterns of electricity moving really fast, how do you change them, right, to essentially have a therapeutic on the other side? In the cardiology case, we have a pacemaker, but how can you create a similar sort of approach for psychiatric illness? And one that scales to the size and scope of illnesses, right? We certainly saw the graphs in which things like anxiety and depression are skyrocketing. And so if you have in silico tools based on putting electrodes in the brain, it's just not going to scale to the size of scope of illness in the world, let alone in the United States, let alone something that shows up in our teenagers. And so we got really interested in this question of gap junctions because it's an entirely different method of routing electrical activity to the brain. And in a model organism, worms, C. elegans, people had actually used gap junctions as a way of changing the interaction between brain cells. And the idea is you could take these gap junctions and put half in one cell, half in another, they touch, you essentially rewire the electrical flow through those brain cells, and the behavior of the worm changes. And so we really got interested if you could use a technique like this, actually putting gap junctions between two chemical brain cells and changing the way information is flowing between them. And spent about 4 years essentially engineering a pair of gap junctions found in goldfish. So instead of working like stickers, you essentially had a positive magnet and a negative magnet. And then we showed that you can deploy these in the brain cells of mice, rewire the way information is flowing through them, and actually change the behavior of the mouse. So we're really thinking about that sort of technique as an innovative platform where we can essentially rewire electrical flow through mammalian brain circuits and in a way of ultimately mitigating diseases like depression and anxiety. So I think it's an area that is incredibly ripe for future development.
I agree with what he said about the potentiality. An aspect that I want to insist, right? Because and I don't know how to frame it is rather than how difficult it is for us to incorporate new notions and new concepts, right? In everything that I think that maybe the electrical synapses are contributing to electrical circuits in the brain has been shown in the retina for years already. So there's there are cells that combine both of them, and then by changing the gain of those synapses can reconfigure circuits from night, right, from the daylight to darkness and back and forth. So the proof of principle is there, right? So it should work on a similar way on other cells, but on other systems. But to give you an idea, that was very clear, the importance of electrical synapses in the 80s, right? And because I'm funded by the Auditory Institute of NIH, I was probably the only one that has a grant on electrical synapses in the auditory system. So it took from the 80s to the 2014 to have the first paper, right, on electrical synapses in the cochlear nucleus, right? And it goes for groups, right? From the retina, they move to the development, and then they move to the cortex, then they move to cerebellum, they move to final the auditory. And it's because each of those group of scientists, they are just biased, right, towards investigating their own ideas and what is accepted in the context of that system. And it's a barrier that we have right now. No one can do, but just imagine that we can operate, with different things and breaking just notions and preconceptions and prejudices in most cases.
If I can jump in here for a second, I really appreciate the detailed answers from the speakers, but I also was really reluctant to jump in front of Nathan because I really want to hear his question too. So want to see if we can end my question. Yeah, thanks to all the answers; they were great.
Thank you so much, Jerry. And I want to say thanks to Kav too, because he actually answered a question that Kavita had put into the chat earlier while he was speaking. So thank you, Kav. Go ahead, Nathan.
Yeah, that was a fascinating discussion, and it was deeper than what I was going to get into. So Jerry, I appreciate your question and listening. I've been taking notes on the side to go do some more reading. This is a really interesting area. One question I had was just on the wearables, just coming back to Amina's talk a little bit, but you mentioned two that I was just sort of fascinated by and I wanted to know a little more about. You talked about an electroretinogram, and then you made just a side comment, so it's totally fine if you don't know more about this, but you made a side comment about being able to do a patch where you could measure, or people could measure metabolites out of the brain. I know you can measure metabolites off patches. Wasn't obvious to me how you do that on the brain. Anyway, I'd love a short answer if you know anything about that. It seemed fascinating.
Sure, I'd be able to follow up. So Shen Zhu's lab at UCSD has been developing a paired patch in the ear that records metabolites at the same time there's an ear EEG. So the metabolites right now, I think, are limited to lactate and glucose, but he's not the only one. So in collaboration, I'm also working with a lab at UT Health Sciences, Kumar Sharma's lab, and he's focused also on non-invasively measuring metabolites in the lymphatic system. So there's a lot of work. I don't know that all of it's published, but there are some, including from the UCSD.
Thank you.
Thanks, Nathan. Sorry, did I just cut someone off? Sorry. I was going to go to Mauro next.
Yeah, thank you, guys. That was a terrific session. Thank you, all the speakers. I have two questions, one for my compatriot, Alberto, and the other one for Amina. So, Alberto, the first question is related. So where are we as a field to be able to target those gap junctions in the brain? Have we been able to make any progress in order to leverage this, say, to promote brain health? The second question is, would it be the brain, perhaps the base area to look at? Gap junctions we have probably in every single organ. In the pancreas, regulate the synchronicity of the secretion of insulin. You have it in the skin, in the heart. So where is your view of gap junctions beyond the brain?
Yeah. So thank you for the question. I don't know, except for the things that Kafui mentioned about the manipulations in C. elegans, right? So far, and he has those attempts to manipulate it by adding the gap junctions in the brain. There is, for chemical synaptic transmission, there is established pharmacology. Unfortunately, we don't have pharmacology for gap junctions because they're the junctions, which is the communicating mechanism, and I explained what I said that it is a doesn't have a specific blocker, doesn't have receptors. So you rely on things that block channels, that block other channels. So all the drugs are dirty, and they have to be very careful when you use them. So unfortunately, there are no tools. In the heart, again, the people have been developing peptides, right, that they can interfere, because that actually, when you have a heart attack, there is a rearrangement of the gap junctions, and that leads to arrhythmias and death or whatever. So there is, maybe that's the way for the future, but right now we don't have it. So gap junctions in general. So yes, we know that the gap junctions are formed by the connexins. And there are family 21 genes. Virtually every tissue of the organism has them. But you wonder that at some point, I mean, and I wonder, maybe a lot of people just work on the channel and the properties of the channel and the cell biology of the channel itself. They might seem all different, right? My perspective is, which is also some sort of similar views in the heart, for example, that the gap junctions that they are in the brain, neuronal gap junctions, are different. So they're mainly formed by one type of connexin, the connexin 36, which only form channels with each other. It's very selective. It's neuron specific. It has a couple of other expressions in other cells, but it's very specific. And if you look to the electron microscopy, it's very clear, that's things that Sotelo brought up in the 70s already. There is a synaptic densities in electrical synapses in neurons that you don't see in postsynaptic densities, very much like in the chemical synapses that you don't see in other cell types like astrocytes and other things. So we believe that the gap junctions themselves are perhaps different. Maybe they're regulated by a molecular scaffold, that we're very interested in identifying all those molecules that regulates them. And it seems to be just even ultrastructural, different from that perspective. And for example, we have mutations of some scaffolding molecules that really show that a scaffold is more important than the channel itself, right? Very much like we call the electrical synapse density in analogy to that. Even further, what is an electrical synapse? It's a definition we are missing, because we say, well, whatever gap junctions are. But you don't define a chemical synapse whatever glutamate receptors are, because glutamate receptors are in non-neuronal tissues as well. So we are trying to actually have proposed that there is maybe other structures like hidden junctions containing ecahedrins and others that forms a synapse that operates with multiple, not only one gap junction, multiple gap junctions, forming structural complexes which are much similar to a chemical synapse and even an immunological synapse. If you just see from that perspective, there is a lot of commonality. So that's where we're working on, trying to address that. But no every gap junction in every tissue might be the same. And there's a similar line of thought in the heart as well, in which the connexin 43 is combined with potassium channels or whatever to form a completely different communicating structure.
Thanks. Thanks so much, Alberto. I want to just quickly, because we're coming up to the end of our time, and I will defer to our Academy's colleagues if I know we have some buffer time to go a little over 2:30. Dacia had a great question about for CAF about how to ensuring access to treatments in rewiring the brains for higher functionings, etc. And I want to thank also Carrie for responding to that in the chat, recommending Nita Farahani's book, The Battle for Your Brain, where she addresses some of those issues of emerging neurotechnology for performance enhancement and considers access inequity issues. Okay, so Claire, I saw you had your hand up and you put it down. Did you change your mind and would like to more advice? I'll go to CAF.
Yeah, go ahead to Dr. Teresa. Thank you.
Okay. Go ahead,
Yeah,
Kyle.
I just want to make a quick comment to answer that really important question that you raised. So I'm also here representing the Brain Initiative on my side job. I'm on the multi-council working group. And so one of the major thrusts that Kari mentioned is an effort to understand gene expression profiles of single cells in the brain. This is both of the neurons and the non-neuronal cell types, of which there are about 200 billion cells. And the idea is if you can understand gene expression profiles, you essentially have a GPS system where you could deliver proteins to those individual different cell types. And so one of the things that is particularly important here, if you think about the landscape of getting gene expression profiles, is that there's a long history of looking at gene profiles and gene expression profiles in both tissue and more largely just looking at gene profiles across the population. And one of the sort of oversights that have been made in the past is that these gene profiles or gene expression profiles haven't always been sort of characterized across people of all different ancestries, right? And so this was a really important thing for us to think about as we were taking the Brain Initiative on and developing HumanNet Cell Atlas. It was to make sure that the cell atlas, sort of what they called generalized. In other words, the same patterns could be used across different people and individuals, different backgrounds and ancestries. It would work across different sexes as well, so that any therapeutic you came down the line would work for men and women and folks of all backgrounds. So this is something the Brain Initiative has been thinking centrally about. A group of scientists, myself and a group of scientists at the Lieber Institute in downtown Baltimore have been working for some time now also to look at some post-mortem brains from individuals of African ancestry to see if there are differences in gene expression across different cell types that may help sort of tune this GPS system. We recently published that work, came out last Monday, in which we can see differences in gene expression patterns that correlated with increased risk for autism. It sort of mapped onto microglia, decreased risk for Parkinson's disease, and decreased changes in gene expression profiles on the epithelial cells, which sort of line the blood vessels in the brain, that sort of also gave correlations for stroke risk. So I think there's a lot of work to do in this area, but it's a really central thing that we should be doing now to make sure that any technological innovations we come down the line apply to improving health for all. So I think it's a fantastic question, and I appreciate you bringing that up.
Thanks so much, Dazi, for the question, and Kyle for that fantastic response. I think Kavita wanted to have an additional question for Gabe. You talked a little bit about international identifiers, sort of global identifiers, but what about sort of in the international context? Are there any norms or standards in this space for these things that we're talking about? Data standards, et cetera. Kavita, have I answered your question, asked your question correctly?
Yeah, it's good enough. Just wondering if the norms and the practices are how they're shared, whether they're shared internationally.
Yeah, I think that just to use as an example, groups like the Psychiatric Genomics Consortium, which have more than 800 researchers from more than like 40 countries contributing data. They've been, they have had to deal with a lot of these challenges that, well, there's different regulatory systems, right? And so that can definitely create a challenge. So you may have certain guidelines that we could put together, about ethical norms that we think as an international community that would be ideal to follow. But there's always the challenge that each country will have its own set of laws, etc. And so figuring out, I think it can help. And whenever we've talked about things like that, I think it can help when you have guidelines like that, to kind of have an example of what a group of experts, at different stakeholders think should be, the way a particular issue should be managed, and maybe researchers from those countries and other people from specific countries can, help lobby in their countries for certain guidelines and regulations. But I think that the more that we continue sort of expanding these efforts of bringing together data sets from from different groups, etc., we'll have to deal with this more and more, and it'll be a challenge. Yeah.
Great, thank you. Okay, so we're just 3 minutes past our end time, and I want to be conscious of people's time. So we did not get to dual use issues today, but I think Kavita might be me saving up some future discussions in that space. So I'll reserve my questions and turn it back to the chair and to the National Academies for any of their closing questions or remarks. But I will say, this hour, this one investment of time for me has been incredibly educational. So I personally want to thank all of these speakers. Fantastic work, each and every one. So thank you.
And thanks to you, Diane, for great moderating. I think the only word for these talks is really, I'm in awe of what's been accomplished. And it's just the prospects of where all this can go. Struck by where you're all taking this work, and the talks were just terrific. Thanks to each one of you for presenting today. We will have a lot to think about. Our board will see what we can come up with to make sure that there's a benefit from all of your work to put these presentations together, and we can carry it forward in some way. Kavita, any comments?
Yes, just want to say thank you all for participating today. This was absolutely fascinating, and so interesting, so many questions, not enough time, but we would like to follow up, certainly. So this is the end of our public discussion for our board members and Health Science Policy Board colleagues. We have a 15-minute break or 10-minute break at this point, and to rejoin in a closed session in 10 minutes. Thank you so much. Thank you all.